Let's start with today's presentation. This is evolution of microscopic imaging uh, from fluorescence microscopy to mass cytometry. So this mass cytometry is actually the imaging version we are going to discuss today. Uh, you might have heard of mass cytometry, proteomics. So this is a kind of imaging. So let's start with uh, let me introduce you. So I am Devashish Sahu. I am currently working as research scientist in New York University School of Medicine at Department of Orthopedic Surgery. Okay, so let's start with the classification. So microscopy till date it has evolved a lot. Hello.
Hello.
Uh,
Sir, you
Microscopy.
are audible. Yes, sir, you are audible.
So till date, it has evolved a lot, and we have many microscopes to discuss about. But today we are going to discuss some of the aspects of how the colorful imaging is done. So you might have seen so many colorful images in seminars, presentations, and I'm basically today I'm going to focus on that particular aspect of microscopy, like what is fluorescence microscopy and how we move forward um, in improving the images where the resolution is more important for getting the proper contrast and getting the proper images. So we'll discuss it through the lecture. So I should not go into all the details of this microscopy, like optical microscope, electron microscope, scanning probe microscope, and imaging mass cytometry. So why I have put it in another category apart from these three categories is this mass cytometry is not a true microscopy. Like it does not involve the principle which we have been reading through. So this its principle is entirely different, and it's although we get the images very similar, or you even cannot differentiate whether it is microscopic image or is a cytometric image. So that is why I have put it in microscopic. So let's go through this optical microscope. So all the fluorescence microscopy, they are a kind of optical microscopy. So compound microscopy, simple microscope, and let's not go into much detailing of this because we have many more things to discuss. So let's start with simple microscope. You might have seen the magnifying glass. So it can be considered as a microscope or simple microscope. So if we call it a microscope, it doesn't sound okay to you. But if I show this picture to you, so this is nothing but a simple lens or magnifying lens put in a dissecting microscope, so that you can dissect your insects or whatever smaller objects or flowers, whatever you want to dissect, you can dissect it through this magnifying glass. And here it is a yeah, you should have gone through the basic principle how a lens is used to magnify an image. So the object is here, and you find a magnified image of this object, and this is eye. You must be knowing that you have a lens in your eye as well. So although this is a single lens, but there are two lenses involved in this. Okay, so let's move on to compound microscope. So uh, uh, so one of the object images of this object, it lies somewhere within the focal length of the this lens. So you need to adjust the lenses in such a way that the object is magnified 2 times. So when it is magnified 2 times, you will get a much more magnified image. And the magnification which you calculate is actually the multiplication of these two magnification. I mean the magnification of these lenses. Okay, so let's move on to our fluorescence microscope. So fluorescence microscope is nothing but it's a it's an extension of the compound microscope or the light microscope, which you often say. So in this fluorescence microscope, you have what extra you have. You can even use it as a light microscope or optical microscope, and without using any uh, fluorescence filter or fluorescence device integrated into it. So in this, what you do is you illuminate a sample, and the sample is it contains some fluorescence tags. I'll discuss it further. And these fluorescence tags emit fluorescence, which is of a lesser energy or more wavelength, and it is then captured through the emission filter to the with the detector. Okay, so the detector can be your eye or it can be your camera mounted over the microscope. Okay, so let's move on to how is it different from a conventional microscope. It uses much higher intensity light to illuminate the sample, okay, as compared to the conventional microscope. The light excites fluorescent species in the sample, which then emit light of longer wavelength. You all know E is equal to h nu or E is equal to hc over lambda. That means longer wavelength is always have lesser energy. So it's if you emit if you hit the sample with a blue light, you may get a red light or a red emission. So that is what we capture, and we can image the object, whatever is fluorescent. So a fluorescent microscope also produces a magnified image, obviously, because it is a extension of a compound microscope. Okay, so let's talk about what principle lies behind this fluorescence microscopy. So you all know, you might be remembering the structure of atom. It contains different orbits having different electrons. So the orbit, uh, the electron at its resting state, when hit by a energy, light energy, or small wavelength electromagnetic radiation, what happens is this gets excited and it moves to the higher energy state, but it is not able to stay there for longer period of time, and it comes back to its resting state. And with this transition of the electron from one orbit to other, there is a emission of energy. So once the first one is the absorption of energy, the electron goes to the higher energy state. Then the electron comes back and it emits energy, which is obviously of lesser energy, and so hence higher wavelength. Okay, so this is known as Stokes shift. So you can see that the energy which is emitted when the electron comes back to its resting state or the ground state, it it may be in the form of heat, and the rest of the energy is emitted uh, as a fluorescence light radiation. So this fluorescent light radiation is then captured using the detector, Okay, so this is known as Stokes shift. So whenever a smaller, so the first one, the first peak, you can see this is the absorption spectrum. So in this, the energy required is of higher frequency or higher energy, and the second, the when the electron comes back to its resting state, the energy emitted is of lower energy or higher wavelength. Okay, so there is obviously an overlap between these two peaks. We can discuss the significance of overlap of these peaks and their significance in the resolution. Okay, so this is nothing but the extension of Stokes shift. I can show you uh, what kind of fluorescent tags do we use, such as the fluorescein, rhodamine, and hochstein. So you can see uh, the excitation wavelength is always lesser than the emission wavelength. So if we uh, tag our protein, where is the protein? The protein is on the cell. Okay, so the protein is on the cell or in the cell, inside the cell. It may be anywhere. Okay, or inside the nucleus as well. Okay, so if that protein which is tagged over the antibody, okay, so the antibody is directed to a specific protein which is localized in the nucleus or which is localized in the any of the organelles in the cell. So this fluorescence is uh, hit by a wavelength of 490, and the emission which is received is green light, which is 520. Sorry. Okay, so uh, sorry for this slide. There are a few things missing here, but I can explain you that if this is a cell and you target a protein, so this protein is actually labeled with an antibody. This antibody uh, is targeted by an antibody, and this antibody is labeled by a fluorescent tag. Okay, so it may be a primary antibody tagged by the fluorescent tag, or it may be a secondary antibody depending on your kind of experiment you are performing. Okay, so what is the significance of this step? So the protein, this is a very specific experiment. The protein which you are targeting, it should be very specific. So that's why we are using antibodies. Okay, antibodies are going to tag the protein. So here you can see that we are differentially staining the microtubules, okay, inside a cell. So some of the parts, some of the proteins are tagged with the green or um, uh, that means the microtubules are tagged with the green dye, whereas the DNA is tagged with the blue dye, so that you can exactly locate where the nucleus is and where the microtubules are. Okay, and you can find out the process or you can image the mitosis of a cell. Okay, so this is an image of the fluorescence microscope. I hope you have understood how the fluorescence microscope works. And uh, we in this lecture we are talking about the evolution of the microscopic imaging. So there are still much more improvement which needs to be done. So why these improvements needed? So there are a few things which are lacking in the fluorescence microscopy. Or to be very specific, we can call this fluorescence microscopy as wide field fluorescence microscopy. Okay, so you can see there is a poor contrast. If you look at this, you may not find it poor, but sometimes when you compare it with a better image, you can find that the contrast is not that good. You can you cannot even locate where the microtubule is. It looks like a bunch of microtubules put together, or a rope. Okay, not a thread. It looks like a rope. Okay, so which is not exactly the case. Okay, now the second is the photo bleaching. So uh, in like I have experienced it myself. If you look at a particular spot on the microscope, the cells will lose its fluorescence after some time. So if you have found a very good image and you have not captured it like immediately, you will lose the image and you have to search again for a better image. So the photo bleaching is really a problem. And there are other things which may be a human error or the instrumental error, but these two, poor contrast and photo bleaching, these are very, very inherent with the fluorescence microscopy. The others are improper excitation or emission filter, sample does not fluoresce, exposure meter incorrectly set, background not clear. So all these things, uh, background is not black enough. So all these things can be managed, but not these two. So what to do about it? Isn't it going further? Yeah, yeah. So let's move on to confocal microscope. So this confocal microscope may be an answer to our problems. Okay, so this is how the confocal microscope looks like. Uh, Today's microscopes are not only a binocular microscope and you just look at these cells and leave it. You need to capture the image. So these images are very, very important for the studies. And let's talk about this, comparison of fluorescence microscopy and this confocal microscopy. You can see that obviously you can see the difference in the image quality. In confocal microscope, you can see the each and every point of the cell is distinct, whereas you have to put very much effort in order to find out the exact points where these green dots are. So this is actually a localization experiment, or the green dots may be some of the proteins which are fluorescing inside a cell on the organelle. So sometimes it is very critical to find out whether these green dots are distinct or not. So what is the difference between the quality of image? What is the reason that the quality of image are very much different? So the reason is the confocal microscope, as the name suggests, is uh, the confocal microscope is very, very specifically using a laser beam. The light source is laser. You must be remembering that I talked to you about the UV, or the low small wavelength lights like blue and far UV, which are being used for excitation of the fluorescence, wide field fluorescence microscopy. But here you are using the laser beam. Okay, so the laser beam is being used to excite the fluorochromes put inside the specimen. Okay, so you can see that in the confocal microscopy, the specimen, the point is very precise, whereas in the wide field, it is uh, it is exciting the entire cell simultaneously. So if this is a single layer of cell, it's fine. If it is multi-layer of cells, so there is a problem with the wide field fluorescence microscopy, but not with confocal microscopy. And you can see that the detector is receiving it's excluding uh, the rays which are coming, which are not focused, whereas in the wide field fluorescence microscopy, all the rays are being received by the detector or the eye. Okay, so one of the things is the UV lamp, here the laser lamp. In the confocal, the specimen is very pinpointed, uh, uh, scanned, whereas in the fluorescent it is not pinpointed. So you can see the difference in the right hand side. You can see that if the cell layer is not unilayered, you can see there are so many cells put together, and the multi-layer of cells, it uh, restricts our vision of a distinct cell, whereas in the confocal microscopy you can see even though there is a multi-layer of cells, you can see a distinct layer of cells, and which helps us to distinguish each and every cell differently. Okay, so here you can see that a single cell, it can even be scanned at different time points with the different laser systems. Okay, so a singular cell, you might have seen the MRI or CT scan. Uh, if you ever go through the MRI and CT scan uh, videos, you will see that the entire organ, whatever organ has been captured, you can see the organ layer by layer. Okay, and there are many images of the organ, so the same organ is being scanned layer by layer, and you can see the different images of each layer. So in such a way that as if it has been cut in transversely or longitudinally into many sections. So this is how the confocal microscope works. Okay, so you will now find out the difference between the confocal microscopic image and the wide field fluorescence image. Okay, so you can in the right hand side, I have shown you how the wide field uh, illumination is done. So the illumination is not that pinpointed, whereas in confocal, the illumination is very much pinpointed. Okay, so you need not scan the entire view at once. So so you can, that is why the resolution has been increased. Okay, so here we are not talking about the magnification. You can see the magnification of both the images is same, but what counts here is the resolution. You can see that the resolution is too much, and you can see the distinct figures here, all the distinct images, and you can figure out where the image is and what is the image is. Similarly, here is a pollen grain. So now I can show you that in a confocal microscope, the pollen grain is has been scanned from one end to the other. Okay, so the 1, 2, 3, and 4, and so on, 12. These are the different sections of the pollen grain. Without cutting the pollen grain into 12 different sections, you can actually see the pollen grain through it. Okay, so here the pollen grain has been started. Here the midpoint of the pollen grain, and here the pollen grain is like it is getting away, and you can see that this is a three-dimensional image of the pollen grain, which you won't be able to uh, you cannot take such image with the wide field uh, fluorescence microscope. Okay, so three-dimensional volume renders from confocal optical sections. Okay, let's look at some of the advantages and disadvantages. I'll quickly go through it. The advantages are obviously reduced blurring of the image. You have seen it yourself, and optical sectioning of thick specimens detection uses highly sensitive photomultipliers, improving signal to noise ratio, and so on. Whereas the disadvantages uh, are still photo bleaching is there, and what more important is diffraction barrier. Okay, so why I have colored it separately, because our next technique is uh, based on this diffraction barrier parameter. So what is diffraction barrier? So let me first tell you that the diffraction barrier is it renders a limitation to the confocal microscopy. Okay, we have seen so many good images that the pollen grain has such a good image. So let's talk about a little more magnification. Then the resolution comes into play. If we magnify a little more, then we have to struggle with the resolution part. Okay, so the if we move further, let's say there is a bacterial cell, and we have to find out the structure of the flagellum, then there is a problem. If there is a cell and it has a nucleus, it is okay. But what if we are moving towards the nuclear pore complex? Then it is a problem. So there the resolution comes into play, and the magnification, you will see that the magnification is almost same. Okay, so let's talk about the diffraction barrier. So if we are using the light source, uh, simple light source, and we are trying to illuminate the cells, all the cells simultaneously, so you can see that there is a 200 nanometer of diffraction barrier. That means you cannot distinguish two different objects beyond 200 nanometer. You can only, so you can see that there are 2 points which are 200, which are distinct, but you can only see these two as two different when they are 200 nanometers apart. If they are not, you can see them as one. So similarly, you will see that there are different peaks, two different peaks with a good resolution. Whereas when the resolution is not good, you will see a single peak for two different objects. Okay, so that means here comes the uh, resolution parameter into play. We the solution is the super resolution microscopy, or the stimulated emission depletion microscopy. Okay, which fetched Eric Betzig, Stephen Hell, William Morner the Nobel Prize for 2014. Okay, let's talk about the STED. What is the how the STED works? So the idea is to, I mean, the objective is to reduce the 200 nanometer diffraction barrier. That means we have to peek into the different points within the 200 nanometers range. So how to do this, we'll talk about that. So this is a detector, and uh, you have two different laser systems. Okay, so stimulated emission and depletion microscopy. So one of the points, so there are two different laser systems. One light is going to stimulate the 200 nanometer area, and the other is going to destimulate or uh, uh, limit the emission, but in a very structured way. So before moving to that, we are talking about the resolution limits. So this shows the resolution limits of human eye, which is 100 micrometer, light microscope, which is almost 50 nanometers, and the electron microscope, which is around 2 angstrom. Okay, uh, so this is around 200 nanometer, the resolution limit of the light microscope. Less than 200 nanometer. So what if we increase the resolution limit beyond 200 nanometer to almost around 5 nanometers using the light microscope? So previously it was perceived as impossible task, but with the advent of super resolution or STED microscopy, you can figure out two different objects placed within the 200 nanometer space. Okay, so here, if the focus of the light is not that narrow, you will feel the two different lights as a single peak. That means you cannot distinguish between these two objects as two. Whereas with the advent of STED microscopy, you can figure out that this is a different molecule than this, because these two different molecules are giving two different peaks. So let's talk about its mechanism. How does it do? So the excitation and the de-excitation. So the de-excitation. So this is uh, an area in which the de-excitation light is being incident over a 200 nanometer diameter of space. Okay, and what about this green spot? So this is the green spot, which is the excitation light. So we are trying to glow. It's like if you put a spotlight in a crowd, if you put a spotlight over a person, you can recognize that person in a crowd. If you put the uh, all the lights in the hall, you cannot distinguish a single person, because the light is illuminating the hundreds of people at a time. So you are not able to distinguish which person is of importance. But if the entire hall is dark and you are putting a spotlight over a person which is of importance, you can easily recognize that this is the person, this is the important person which we should look for. So this is how it is actually putting a dark, I mean, it is actually de-exciting the molecules around the single spot. Okay, so it so this is known as de-excitation. So here it shows I should correlate this image with this image. So the diffraction problem or the diffraction limit is here 200 nanometer. So I am showing this 200 nanometer area. So this 200 nanometer area, so it is now the midpoint is being spotted. Okay, and the rest of the part is being de-excited, so that the person of importance is illuminated and it has to be captured in the camera. So what we do is we give importance to each and every molecule one by one. Okay. So for us, every person is VIP over here. Every molecule is VIP. So, but we have to give importance one by one. Okay. So this is known as keep some molecules dark. So this is the solution. So what was the problem? Molecules within less than 200 nanometer diffraction barrier are not discernible. Okay. So now we have got the entire image. Okay. So now you can see the difference in the image of confocal and stead. Okay. Look at these two molecules. When you look back in confocal, these two molecules are considered as one. But here you can clearly see that these two molecules are different. Okay. Similarly, this is I was giving you an example of nuclear pore complex. So the nuclear pore complex, you cannot visualize the nuclear pore complex using confocal microscope. Okay, whereas the STED, you can easily distinguish the 8 different molecules comprising or which make up the nuclear pore complex. Okay, so this is how you have clarified the object even more. The resolution is much better now. Okay, and what else? You can visualize the neurophysiology of a live mouse. Here, a mouse with a skull open, and obviously the neurons, they are emitting some fluorescence because they are somehow tagged with the YFP. So the fluorescence emitting neurons, I cannot show you the video here. Uh, I would love to give the video here, how the neurons at a very real time, you can see its movement, you can see how they behave uh, under the microscope. Okay, so it was not possible before the advent of super resolution microscopy. Okay, let's talk about, so since we are going through the evolution of microscopic imaging, so all these microscopic techniques have their own advantages and their own limitations. So whatever we have talked about, they hold importance till date. Okay, so they have not lost their importance. With some advantages they have and some disadvantages, we are trying to find out the next possible microscopic technique. So now we are even talking about the limitation of super resolution microscopy. So what are the limitations? The limitation lies in the fluorescence itself. Okay, so the use of fluorescence is the limitation in of the microscopy. So for example, let's talk about the uh, the nuclear pore complex. So there are 8 different molecules. What if we are trying to scan the cell membrane, and which consists of more than 8 different molecules, or 8 there are so many proteins, not only 8, there are hundreds of proteins. You want to scan each and every protein separately. What will you do? You have a limited number of labels. 3 to 5 labels are there only. So how you are going to tag each and every protein of interest with the 3 or 5 fluorescent tags? So this is a problem. And the second problem is the, uh, there is a gap. I talked about this gap. The gap, sorry, not gap, this overlap. So I talked about this overlap of the absorption and emission spectral profile, and this overlap is a concern here in this case. So what does it do? it will further hinder our distinction between two different molecules as far as the tag is concerned. So let's talk about the fluorescent tag. What is, how does it uh, create a problem? So let's, uh, you might have seen the plasma membrane. So this plasma membrane, you can see that there are different proteins which are getting tagged with the different fluorescent tags. Okay, so let there be so many fluorophores. So what the problem is, still the overlap. Okay, then what is the solution? Replace the fluorophores with the metals with and do atomic mass spectrometry and imaging atomic mass spectrometry or imaging mass cytometry. Okay, so what is imaging mass cytometry? It is a highly complex imaging technology. So it is quite newer than rest of the methods. And in 2014, they have developed it. And after 2014, there have been many modifications and many improvements over this imaging mass cytometry. So let's move through this technology. So this is a highly multiplexed imaging technology, has the same advantages over traditional fluorescent microscope that Helios has over flow cytometry. Oh, okay, so there are a few companies like Helios, Fluidigm, which deal with the imaging mass cytometry. The patent is still with them, and they so not many companies prepare or manufacture uh, this instrument. So again, there is a team laser, and this team laser is incident on the object or the cells, okay, of our interest. Okay, so this is just an overview. The story remains much more than this. Okay, so these are the labeled cells. So here we are not labeling the cells with any fluorescent dyes such as FITC or rhodamine or something like that, or Cy5 or Cy2 or Cy3. These are the fluorescent dyes which we normally use in case of flow cytometry or fluorescence microscopy. So here we have replaced the fluorescence dyes with the heavy metals. Okay, so I have shown you why these heavy metals. You can see that the peaks are much distinct. They don't overlap with each other. Okay, and the why heavy metals? Because normally the heavy metals are not the component of the living system. Okay, so these labeled cells are burned with a laser. Okay, so what happens if it burns? We have destroyed the cells. But the answer lies with the metal ion. The metal ion still remains there. It has nothing to do with the burning. It withstands the burning. So the metal ions are formed, and these metal ions are accelerated into the mass spectrometry or time of flight mass spectrometer. So uh, these cells are burned in such a way that these form a packets of metal ions. That means a metal ion of a single cell will be different packet, and the metal ions of the subsequent cell will be a different packet. So a single packet is processed at once. Okay, so you can, that's why you can find out the composition of the cells tagged with the metal ions. Okay, so you might have seen that the metal ions, when they are accelerated through this quadrupole, they move into the mass cytometer or mass spectrometer, and they are accelerated, and they are subject to a time of flight. And you must be knowing that the time of flight is very much important in order to find out the uh, difference in their uh, in their mass. Okay, and the mass corresponds to the protein which we have tagged. So we have deliberately tagged a particular protein with a particular metal ion with a specific mass. So that specific metal ion corresponds to that protein. So these peak intensities will so this set of peaks will show that there is a set of proteins which are associated with a particular cell. Okay, so metal ions have very little overlap and lots more channels. Okay, so almost 32 different proteins you can tag at once. Okay, so if you want to find out more than 32 proteins, so you can run different um, uh, set of experiments of the same set of proteins. So you can find out even more than 32 different proteins. So it will be much lesser tedious than the fluorescent microscope, which use 3 to 5 fluorescent tags. Okay, so in this, in IMC or imaging mass cytometry, you have to prepare a very thin layer of cell or tissue, and you have to tag all the proteins of interest with the metal ions. Now, once these are tagged, washed off, you have to burn the metal, the cell layer uh, with the UV laser. And uh, once it is burned, there are packets of metal ions which are flown into the mass cytometer. There are different signal extraction of measured markers, 32 measured markers. So all these panels, they are put together. They make up the entire image. Okay, so you can imagine how interesting this technique is. You are burning out the cells, still you are getting the image. Okay, image out of the ashes. Is it so? Okay, so single cell segmentation and downstream data analysis. Okay, so this is how the so this is a picture how the if we do not intend to find out the image of the cells, we can use it otherwise in order to find out the for example single cell proteomics mass cytometry. Okay, so this entire image shows that how the single cell proteomics is done. So I should not go into the details because the principle remains the same. Just uh, the difference is there you were using a sheet of or a thin layer of cells or tissue. Here you are using the cell suspension. That's it. That is the difference. Here you are not reconstructing the image. Okay, so the advantages and applications of imaging mass cytometry. Metal labeled antibody cocktail targeting specific antigens of interest under resting or stimulating condition. The cells are fixed prior to mass cytometry acquisition to preserve the cell and minimize variance in the mass spectrometry. Detector reduced sample to sample signal variation. The time resolved detector measures a mass spectrum that represents the identity and quantity of each isotopic label. Yes, this is also one of the important things, that fluorescence microscopy is not quantitative, whereas imaging mass cytometry is a quantitative technique. There you for quantitation you do fluorescence cytometry. Single cell imaging with antigen quantitation is possible. Okay, so this is one of the very latest techniques, single cell imaging and single cell proteomics. Okay, so this is possible through mass cytometry. But what are the limitations? We cannot take images or videos of live cells, uh, just like we are doing it with a super resolution microscope, and we cannot retrieve the cells for further analysis as the cell components are burned out. Overall resolution is lesser than super resolution microscope, so we are more concerned about the different types of proteins which we are interested to study. So that is why here the resolution and the gap in the resolution is not that much. So I would like to hear some of your questions if